Oh, wow, yeah, that isn't working great. Okay, so my name's Nick. I'm a group leader in assay biology here, and I'm going to be telling you about how we use biophysics for lead discovery. So it won't come as news to anyone in this room that cutting-edge drug discovery is embracing a whole new series of modalities, and that can range from targeted protein degradation, ADCs, and cell and gene therapies. But today I'm going to be talking to you about how Domainex approaches molecular glues and targeting RNA. RNA-directed therapeutics aren't a new field per se, but the way that people are targeting them is changing. So traditionally, the two main approaches were siRNAs and antisense oligonucleotides. siRNAs are small, interfering RNAs. They are short, double-stranded sequences of double-stranded RNA that you can design to be complementary to your target of choice. siRNAs work by, if you administer them to your patient, they enter the body, they get into your cells, and at that point, they hijack your antiviral machinery. They manage to do that because a lot of viruses have got double-stranded RNA genomes, and so they can enter the gene silencing pathway through Argonaut, through RISC, and then binding to DICER. And because you've designed your RNA to be complementary to your target protein, it can very specifically degrade the RNA of that protein, reducing the levels. So it's a really nice, highly targeted approach. And there are quite a few siRNA drugs on the market. These are just a couple of examples. Onpattro, Givlaari, one of which is for an amyloidosis and the other for porphyria. The other main approach has been antisense oligonucleotides. So ASOs are modified RNAs. They're not actually RNAs themselves. And the reason why they're modified is that RNA has a couple of main drawbacks. It's very unstable, particularly when you put it in the body, and it has pretty poor cell penetrance. They tend to be larger molecules, quite highly charged, so they struggle to get across cell membranes. And so by chemically modifying the backbone, you can improve both stability and cell penetrance. When I started looking at these, I assumed that most ASOs were actually translation blockers, but they're not. Most drugs on the market are actually alter splicing. I think probably the most famous is Spinraza, and that is a treatment for spinal muscular atrophy, which is a pretty grim degenerative disease that can shorten the life of children to just a few years. SMA is caused by mutations in the SMN1 gene, and Spinraza works by binding to the mRNA of its homologue SMN2, and by binding to the mRNA, it blocks a splicing site and forces the SMN2 gene to include an exon that makes it functionally the same as SM1, and so it can escape this genetic defect. Vyondys 53 does the opposite. It binds to a splicing site in dystrophin and forces it to exclude exon 53, and then it alleviates the symptoms of DMD. As I said, though, using RNA has got a few different problems. It is unstable, it's got poor cell penetrance, and it can't get through the gut wall. Luckily, med chemists have got decades of experience of designing compounds that are stable in the body, get through the gut, and get into your cells. And so using small molecules to target RNA is becoming more and more attractive approach. And this isn't totally new. There are quite a few antibiotics that target ribosomal RNA, so things like streptomycin. There are also other anti-infectives like remdesivir, which is a treatment for Hep C and been used a lot for COVID. And I think of particular interest is risdiplam, which is also a treatment for SMA, spinal muscular atrophy. And not only is it orally bioavailable, but it actually has got slightly fewer side effects than Spinraza, so it's not just route of administration that can be improved by using small molecules. One of the things that's held back RNA-directed small molecule drug discovery has been essentially concerns about druggability. So when we think about RNAs, we often think about linear strands floating about in the cytoplasm, waiting to bump into a ribosome. But actually, RNAs are much more structured than people might think. This is tRNA, and we've probably all seen stem loop diagrams like this before that sort of give you an indication of the structure of RNA. And they still look pretty simple, but when you look at them in 3D, that RNA is far more complex than you might think. And while that's tRNA, mRNA has also got a lot of structure to it, a lot more than you might think, particularly in the 3' UTRs where you've got stem loops, hairpins, and quadruplexes. This is just risdiplam bound to mRNA. And on the right-hand side, we've got the SAM6 riboswitch. So a riboswitch is a structured RNA that bacteria use to bind to metabolites and control their metabolism. So it might not come as much of a surprise that the SAM6 riboswitch binds to SAM, and this is the tool that we've used for establishing our RNA-based platform. One of the challenges of working with RNA is they often don't have an easily measurable function, like you can't just run an enzyme assay. So this is where biophysics becomes essential in your development process. Biophysics-based binding assays can allow you to identify your binders and develop them. And luckily at Domainex, we've got a really comprehensive suite of biophysical assays. We were early adopters of the nanotemper technology, so MST, but we've moved on to their latest generation of instruments, the Dianthus, and on that we can use both trick assays and spectral shift. We use spectral shift a lot, it's really great. We were also early adopters of GCI, which is an SPR analogous technology, flow cells and chips, the main difference being how the light beam interacts with the plasmon field. For protein thermostability, we have both DSF and nano-DSF. So nano-DSF uses the intrinsic fluorescence of your proteins to look at unfolding, and old-school DSF uses dyes like Cypo-orange. If we want to look at thermodynamics, we've got an ITC, and the technology that we've been using quite a lot recently is mass photometry, which is a really interesting technique for measuring protein complex formation. Does that look all right? Yeah, okay. The first step in building the RNA platform is to showing that we can actually set up some assays. So what we've got is a couple of different assays on this slide. On the right-hand side, we've got data from the Dianthus showing equilibrium binding of both SAM and SAR with much better affinity of SAM for the riboswitch, which is what we'd expect from the biology. And on the right-hand side, you've got GCI, and anyone who's familiar with SPR will recognize this kind of sensogram. And just like SPR, GCI can give you both affinities and kinetics. And when we compared those with the literature data, we're really pleased to see that they were aligning well. And so we think we had the strong basis for developing our RNA systems. And that is all very well and good, but how do these assays operate in a drug discovery setting? So to do that, we used a fragment screening approach just to show that we can. Sorry, this screen is very strange. But rather than just screen the entire fragment library, we took a computationally guided approach with Mike, who's our head of Comp Chem, and you'll be hearing from him lately. He used a combination of computational methodologies that I don't understand to predict SAM6 binders from the fragment library. And so from that, we took a subset of 100 compounds, which we used for a single concentration screen. And that's the data you can see on this left-hand side. This is the single point data which we generated using spectral shift. The nanotemper software is quite conveniently will identify all the compounds that are interfering in your assay and giving you false positives, and then just leaving you with the binders and non-binders. And when you summarize that data down and simplify it, it's gone. Okay. We had a hit rate that was about 33%. I don't know, man. Just keeps going off. That we thought was really great from the virtual screen, and so we think this is a really strong platform to build our RNA-directed assay floor map. Right, sorry. So the second half of the talk is going to be about molecular glues. These are small monovalent molecules that drive induced proximity between two proteins or stabilize natural PPIs. Glues have become much more interested in the field recently because they're being seen as a powerful alternative to PROTACs for targeted protein degradation. They've got a couple of they're a bit better than PROTACs in terms of hit finding, and they also overcome some of the physicochemical challenges of using PROTACs. But molecular glues have got applications beyond TPD, such as stabilizing natural protein-protein interactions, and you can see that in this structure here, where you've got a molecular glue stabilizing the interaction between KRAS and PIK3CA. So to accelerate discovery in this space, Domainex has developed a comprehensive suite of assays specifically designed to support the ID and optimization of molecular glues. So we have a cascade or a set of complementary assays which are suitable for different throughputs and all have their different advantages. When we're looking at large compound sets, we typically use AlphaLyser or FRET. These are molecular proximity assays that are absolutely ideal for measuring ternary complex formation, which is exactly what you need when you're looking at molecular glues. AlphaLyser and FRET are well-established assays that scale really well to high throughput, and they're always sensitive, robust, and reliable. And so a big fan. And this is just some nice AlphaLyser data we have in molecular glue. For smaller compound sets, so fragment screening or following up virtual screens, we often use spectral shift. Spectral shift is an in-solution fluorescent assay that has a couple of advantages over AlphaLyser and FRET. In particular, you can do both binary interactions and also ternary complex formation. One of the nice things about spectral shift is also it eliminates the needs for beads or labeled antibodies. So if there's any concerns about steric hindrance in this interactions, you can exclude those using spectral shift. It's compatible with medium throughput, and as I mentioned a couple of slides ago, glues are important beyond targeted protein degradation, and you can see here some nice data where we've enhanced the interaction of a naturally occurring protein-protein interaction. Another one of our favourite assays is GCI, so this is the SPR-like technology. Just like spectral shift, you can use it to measure both binary interactions and ternary complex formation, and just like SPR, you get both affinity and kinetic data showing you your complex formation and it coming apart. So this is very data-rich and great for getting deeper mechanistic insights to guide your compound optimization. When you start getting into the gold standard assays where you're looking for label-free in-solution experiments, we tend to use mass photometry and ITC. So mass photometry is a really nice technology which uses light scattering to literally measure the mass of your proteins. And what you can see in this graph in the top corner here is what happens when you introduce a molecular glue to two proteins, one being a mystery protein A and cereblon. You see this rightward shift of the peak, which is the complex forming. You can't see the smaller protein in because the downside of mass photometry is it's got a 30 kilodalton cutoff. So it's not so great for smaller proteins. When we want to look at different types of parameters, we use ITC because that can give us both affinities and thermodynamics. But of course, that's really protein hungry, so we tend not to use that very much. So in summary, cutting-edge drug discovery is being driven by a host of new exciting therapeutic modalities, one of which I think is targeting RNA with small molecules. One of the challenges of working with RNA is that it typically lacks an easily measurable function, and so biophysical techniques are essential for both identifying your binders and characterizing them. Inducing PPIs using molecular glues is opening up entirely new areas of biology as a therapeutic target, and for any glue program to succeed, it's essential to have a good set of biophysical assays to look at both your complex formation and other aspects of the development. And at Domainex, we've got a comprehensive suite of complementary biophysical techniques that can power any drug discovery program that's exploiting these revolutionary approaches. And that's me. So thank you very much.